So the chapter opens up with a story on this Euterites family. I'm not sure if I'm pronouncing it right. Is that how you say it? Euterites? Families, they came from Austria, and they actually have this phenomenon that's called the settler, or there's a specific name for it, founder effect. Okay, they came as a very small group of people, maybe about 100 people or so, and they settled in one place, and they are a very close group of people, meaning they do not usually marry outside of their immediate family, like cousins or second cousins. So now, after the 1500, after they migrated here, they have expanded to about 40,000 people in some specific states and also parts of Canada. And what happened with this founder effect is that one or two people in the group that originally came, that migrated, had this recessive gene that causes this syndrome, or this disease that's called the Bowen-Conradi syndrome. And what that does is that if you get two of the alleles that have this defect, two of these recessive alleles, you just cannot process translation very efficiently. So what happens is that the person that is homozygous for these recessive alleles, they do not develop normally. The fetus does not develop normally. It's a very small fetus. They are born usually with very small heads and very much a lot of problems, genetic problems, like joint malformation and metabolic problems. Basically, this syndrome is lethal, and it kills very quickly. Within the first year, the child will die if the child inherits both of the recessive alleles. And this group of people is the only people known that carries this syndrome. So when this syndrome was being studied, they know there was very confined to a specific population, and they could find out what happened, what was the gene mutation, and how the syndrome was progressing. So you can imagine that if you have a problem with the translation from a messenger RNA to proteins, it is not a problem of only one system in the body. It's a problem of the whole body, because every single cell in your body will need to make protein. That's what your body is made of, right? Proteins and minerals. Inside your bones, you have proteins. Everything is made up of proteins. So if you cannot make protein, you can imagine they're not very successful in surviving. So now we're going to the genetic code and translation. And this outline for chapter 15. We are going to look into genes that encode proteins, also the genetic codes that will describe how the nucleotide sequence will specify the particular protein, and also on the assembly of the protein and properties of RNA and ribosomes and how it affects protein synthesis. We're also going to take a look at the codes, the genetic codes, and also the idea that one gene will produce one product. Here, just for a brief review on the transcription, we know that we have the DNA that has the code, and you transcribe it into RNA, and from there you have to RNA in the eukaryotes has to leave the nucleus, and in the cytoplasm you are going to translate it into protein. So now we're going to the translation. It happens in the cytoplasm, not in the nucleus. Remember, this is a compartmentalization, one of the main differences between the prokaryotes and eukaryotes. In eukaryotes, you have the nucleus only for transcription, and then the cytoplasm for the translation. In the prokaryotes, you have everything that can happen at the same time. Always keep that in mind, and you probably hear me repeating that about 50 times tonight, because it is a very important concept. So now let's make a protein here. We know that the ribosomes can read the protein's building blocks, and they're going to be involved in the formation of whatever is going to make the protein from the RNA. We also know that mRNA has the ribonucleotides, A, U, C, G, and also that the ribosomes reads the messenger RNA in a linear fashion. It does not just get from one end and jump around and go backward and forward. No, it gets from one end and reads the whole RNA until it gets to the other end or the stop of the end of the protein, and then it will fall off. Now, we know that the RNA is what is carrying the message to make the protein, and the ribosomes are going to translate the message into the actual protein. Now, how is this code, or how, if you imagine the RNA as having the words, how are these words organized? Now, from earlier experiments, even from Francis Crick experiments, they knew that amino acids were on a linear fashion with the messenger RNA, and they knew that the codes for each amino acid will have to be composed of 3 nucleotides. Now, Crick figured out that there were 3 nucleotides that would make one codon because he performed some mutations with his messenger RNA experiments. First, he decided to add 1 extra nucleotide here. He made a mutation on his RNA. He wanted to figure out how many nucleotides are going to make a protein for one amino acid. So he inserted a nucleotide right there. He inserted an extra U, and he decided to see what happened to the sequence of amino acids. So at first he has his original mRNA here, and he figured out to get a certain amount of amino acids. Now imagine that this is like lysine, leucine, valine, in a particular sequence. So he wants to know if he inserted one base here, what will happen to this sequence of amino acids. And he figured out that after 3 bases here, and he inserted one here, he figured out that he can get his first amino acid there, and the rest just did not match with his original sequence. He said, okay, this one is telling me that I need 3 bases to have a translation for one amino acid. He said, well, let me try something else. Let me try to add 2 bases and see what happened to the sequence of amino acids. And there he goes again. He translated. Still, he got only the first one, and everything else did not match with his original sequence. Now he decided to add 3 nucleotides after the original first 3, and then he wants to see what happens to the sequence of the protein, and he figured out that the first one matched, the second one did not match, but then everything else matched with his original sequence. So he figured out he needed 3 bases to get one codon to translate one amino acid. Quite a clever experiment. Now, if you look at it from a different view, you can say, how are these words organized? If you imagine that the DNA is just like a dictionary, or just like the way we use words and letters to make words, and if you can get a lot of 3-letter words together, and you want to make sure that your sentence will make sense. So here we say, for example, if you have here on your mRNA, the dog beat the man, okay, everything is 3 letters, and it does make sense to read the dog beat the man. Now, if you insert one random letter after the first 3, you are going to have a frame shift of whatever else is written there, and if you try to read it in 3-letter words, you end up with the dog beat the EMAN. So you don't really understand what is written afterwards. You try to add 2 letters, and what happens? Again, you have a mess. The THEBDOGBITTHEMAN. So it still doesn't make sense. Now, if you add 3 letters for this codon, it finally makes sense again, just like we did in the previous example. The big dog beat the man. You have the frame that went back into gear, the frame shift here. It was Crick, yes. Yes. Sure.
Now, what is the reason that the second amino acid that text there? Is that because the very last one insert three? So it was one X and one X, so that X is where the insert is.
Or you mean here?
No, the last one.
Oh yeah, this one is the amino acid that did not match. It's just a different amino acid. Huh? This is the amino acid that came as a result of putting these three nucleotides there. And the X is not that all the amino acids disappeared.
Right.
It's just that they were different from the original sequence. It just did not make sense as it's supposed to make sense from the original sequence. Just like the big dog is not having something.
Okay.
It didn't make sense with the amino acid. Now, then they wanted to figure out how this code was organized, because they knew that you have 20 amino acids, and they also knew that you have 4 nucleotides, and how can you organize them in order to get your 20 amino acids. Another evidence that would point to the 3-letter codons would be that if you only use 2 letters, of course, if you only use 1 letter from one amino acid, you're only stuck with 4 amino acids. And that's not the case. If you use 2 letters for a codon, then you're stuck with. Now then, if you use 3 letters instead, so now you have 3 letters and 4 nucleotides arranged in random fashion. That will give you 64 possible combinations. So it can give you theoretically 64 different amino acids. And that's plenty because you only have 20 amino acids that are naturally occurring, around 20. Here is for clarification of the probability for how many bases are needed to have at least 20 amino acids. So here, if you have 1 base, this is just the example that we have, that if you had a bag with stones in it, in this case we have a bag with 4 bases in it, and we are going to pick 1 base at random. So if we only have 1 space, we can pick 1 of 4 bases. If we have 2 spaces, we can pick, in this case here, 1 of 4 bases. In this other case here, 1 of 4 bases, and they are independent terms, so they multiply. And here you can have a total of 16 combinations or 16 amino acids. If we have space for 3 bases, and we can pick 1 of 4 in the first, 1 of 4 in the second, 1 of 4 in the third. Again, you have independent terms, they multiply, and you have 4 times 4 is 16, times 4 is 64. 64 possible combinations for the amino acids. Now, how are the groups organized? We see they are in the 3 ribonucleotides, and those are called the codons. Each 3 letters is going to be assigned for 1 amino acid. And each one of these codons is unambiguous. That means that if you get ATG that is telling you is a codon for methionine, it will not code for another amino acid. It's not going to code for phenylalanine, and it's not going to code for any of the other 20 amino acids that are out there. And also we know that this code is universal, or I like to say is an Earthling code, because we don't really know what happens in other planets. We did not have communication from that side yet, but at least on Earth, this is a universal code. For all the species that are alive on Earth, they do follow these rules for the codons making proteins. Now, how does that code work? If you have 6 different nucleotides here, and you want to figure out, now we figure out that, okay, we need 3 letters for a codon. Now, how is it that it's going to pass the message on the RNA. You could have several things that could happen. You could have this on a non-overlapping linear form. The second 3 nucleotides on one codon is for your second amino acid. Or you could have some type of overlapping codons, which in this case would mean that if you have GUA, that will be your first amino acid. And then you could have UAC for your second amino acid. And then ACG for your third, and so forth. So then in this case, if you have 3 non-overlapping bases, out of 6 nucleotides, you could have 2 codons that will come up, this and that. Now if you have overlapping, then out of 6 bases, you could have 1, 2, 3, at least 4 different codons that will come out of this. Now, with the experiment, if you change the U in the above sequence to a different letter, how many amino acids will you change if it is overlapping or if it's not overlapping? Let's see, if you change this U here, and then you do your translation, you're going to see that the first amino acid was changed to something else, but then the second amino acid did not change to something else. And this is the way they proved that the code is non-overlapping, that you don't have all this mess going on here. Because imagine how difficult it would be for the ribosomes to translate some type of overlapping code into protein, and they will come, read 3 bases, run back a little bit, 1 base, read the other 3, and that's a room for a lot of error. So if you have a linear, non-overlapping code, you minimize the chances of error. Okay, so coming on to the properties of the genetic code. We know that the genetic code is degenerate, which means there is some type of redundancy on this code. So what it means that we know that we have, if we need 3 nucleotides per codon, and you have 4 possible nucleotides per space here, we know you have 64 possible codons that are there. Now, we only have 20 different amino acids. So what happened to the rest of the codons? We have 64 codons and 20 different amino acids. So there are 2 possibilities here. 44 codons, which this is getting by 64 possible ones minus the 20 codons for each of the proteins. It could be the codons code for nothing or for only 1 amino acid, which in this case it would be a wasteful way of organizing your codons. Or it could be that more than 1 codon could code for the same amino acid. In that case, it's a smart way of organizing these genes, because if you have a mistake on one of the bases, you can still get the same amino acid put in there. So now we know that there are studies that show that more than 1 codon can code for the same amino acid. And this is what it means by degenerate. The codons are degenerate. Now, for example, here you have 1, 2, 3, 4, 5, 6 different codons that code for serine. Now, we also know that on these codons, the last nucleotide of the codon is the one that is not very important. Meaning that if you mutate the last base here if you mutate this U to a C, oh, look at that, you still get serine. If you mutate this U to an A, you still end up with the serine. And so far, you mutate this U to a G, and it still is coding for serine. So you do have a little space, a little room for error here in case you had an error in the transcription, or in case you had a mutation on the original DNA. You still can end up with the same functional end product of a protein. And the last nucleotide leniency here, an error,